name is Dr. Taylor Cottle, and I am the CEO and co-founder of Sharp Diagnostics, where we're bringing molecular testing to every home to make accurate nucleic acid-based tests available everywhere in the world. When it comes to our health, it's no surprise that decisive results can save lives, and I'll give two examples here. Where on the left, you see an example of what factory farming looks like. Thousands of animals crammed into tiny, tiny enclosures, and the spread of disease, kind of like we saw for COVID, is actually rampant among these as well. So having a way to test animals for viral infection is actually supremely important for the biosecurity of our global food supply chain. That's the first example. So a second, slightly different example. If you think about bacterial infection, particularly, let's say you have a combatant on the field of war where they're suspected of a blood infection, but the doctor on site only has hours to administer the right antibiotics before this patient goes septic. So it's important to have definitive, accurate results to know what antibiotics to administer. Together, these two examples represent billions of dollars of economic loss with millions of animals killed and millions of infections with hundreds of thousands of deaths just in the United States, not to mention around the globe. So we believe next generation diagnostics should be both definitive, highly accurate, so you trust the results, but also on demand and be able to be accurately performed on the site of care from sample to answer. Now, this is the gold standard of PCR, polymerase chain reaction, but it falls short in several areas. It requires several days to result, requires expensive hardware and a trained technician. And here at Sharp Diagnostics, we're building devices that give minutes to result, use only simple equipment, and have simple operation. But how do we do this? So the technical problem is that traditional PCR uses a thermocycler. It's a piece of equipment that physically ramps up the temperature and down the temperature to split the double strand of DNA to make PCR viable. What we've done is invent an enzyme that allows us to use chemical energy, or ATP, to split the DNA duplex instead of machinery, allowing us to bypass all of the hardware and do the entire PCR reaction at one temperature, isothermally, in a test tube. In brief, the enzyme is essentially a super helicase that can drive along the double-strand duplex and split it in real time. And this is a patent-protected enzyme that has come out of the PhD and postdoc work of my co-founder, Momchila Gavrilov. Overall, this is a robust technology that's been published in a highly reputable journal, and it also, importantly, uses all of the same principles as traditional PCR, which means it's essentially plug and play with the current systems that we have to do general nucleic acid amplification. In short, we can take what takes in the lab 2 hours or several days if you have to ship to a lab. We can translate that process into a test tube result going from sample to answer in around 10 minutes. We envision selling this diagnostic as a swab or blood sample that can be inserted into essentially razor blade cartridge that is compatible with a fairly simple analyzer that requires much less hardware than conventional technology because of our advances in the enzymology behind the technology. Compared to our competitors, the traditional PCR takes days. We're much faster. We're more accurate than the traditional antigen test that you may have taken to see if you have COVID. These are 80–90% accurate, where we can be as accurate as a traditional PCR test. Because we're primarily enzyme-based, we can reduce the costs from PCR testing to be comparable with antigen tests. And we can also multiplex diseases, which, based on customer interviews, is something that's highly needed in this space, being able to take multiple samples, but also multiplex to test for multiple diseases in a single test. Finally, we're point of care, which means this robust test can be used with our particular device at the point of care. Now, our go-to-market path involves a stepwise approach in order to continually de-risk the technology and approach increasingly valuable markets. Where we'll start with the research tools market, which includes mostly biotech companies and academic markets where we know we have inbound interest to use the technology. It's a smaller market, but then will allow us to de-risk the technology into the ag tech diagnostics, which will require either CLIA lab or pre-market FDA or field device generation, and then 510K FDA clearance. This will all provide data to support our final human diagnostics application, which is the largest of markets. And here you can see that the research tool market, though is small, again, allows us that stepwise direction towards the ag tech diagnostic market, which is a significantly larger market representing hundreds of millions, where finally, that allows us to reach the human diagnostics market, which we believe is a significant market that allows us to get into the home care space, which is something that traditional and existing technologies has not been able to do. I'll also add that the research tools market also allows us to approach sub-licensing opportunities, which I think is a significant advantage for us. Traction today, we filed the patent in August of 23 through the Hopkins Tech Transfer, and we're currently negotiating an exclusive licensing. Yeah. We've also raised $300,000 in the last 3 months, actually, and have access to a $300,000 uncapped safe through the support of the Activate Fellowship for their 2024 cohort.
That's great.
We've also 3D printed a field prototype that I'm excited to continue with, and we are looking to raise 1.5 and then Q1 of next year. So our team is made up of the inventors that are behind the base technology that make this whole product possible. My co-founder, Momchilo Gavrilov, was a PhD at postdoc at Johns Hopkins and received his PhD in Canada. And our advisory board includes the scientific brains behind the helicases involved, as well as medical expertise and experts in the life science realm of business as well. Overall, we hope you'll help us reshape how molecular diagnostics are done in bench side, in agriculture, and hopefully bring accurate and reliable tests to everyone's home so that no one has to be unsure of the health of their family. Thank you.